Hi, my name is Brock Hashem, and I would like to welcome you to the 6th episode of Histo Helpers. Our objectives today will be to identify the 3 types of muscle: skeletal muscle, cardiac muscle, and smooth muscle, and to be able to explain the similarities and differences in structure and function between these muscle types. Let's begin by taking a look at slide 217, which is a section of skeletal muscle prepared with H&E. On this slide, we can see 2 sections of skeletal muscle. On the right is a longitudinal section, and on the left is a transverse or cross section. Let's begin with the cross-sectional view to the left. Before we begin to identify structures, I would like to clarify the terminology I will be using. Skeletal muscle cells are commonly referred to as muscle fibers, so understand that these terms can be used interchangeably. Muscle cell membranes are often called the sarcolemma as well. Lastly, muscle cells may be referred to as myocytes. As I zoom in to the 20x objective, the individual muscle fibers become readily visible. A muscle fiber can be seen here at the light green arrow. Immediately surrounding each muscle fiber is a thin layer of loose connective tissue known as the endomysium, which is represented by the yellow line. Groups of muscle fibers are bundled together into fascicles, which are surrounded by a thicker layer of connective tissue called the perimysium. Fascicles are functional units of muscle fibers that tend to work together to perform a specific function. A muscle fascicle surrounded by its perimysium can be seen here outlined in light blue. To reiterate, the endomysium surrounds each individual muscle fiber, and the perimysium surrounds each bundle of muscle fibers, or fascicle. Notice the peripherally located nuclei in each of these skeletal muscle fibers. These nuclei can be found immediately beneath the sarcolemma of the muscle fibers. One can be seen at the tip of the red arrow. Recall that skeletal muscle fibers are large, multinucleated cells formed by the fusion of myoblasts during development. We are now looking at the longitudinal section of skeletal muscle on the right side of the slide. In this longitudinal section, I would like to again point out the peripherally located nuclei of the skeletal muscle. Each muscle fiber is comprised of longitudinal subunits called myofibrils. In turn, each myofibril consists of actin and myosin myofilaments. Myofibrils are organized into contractile units referred to as sarcomeres. 3 parts of the sarcomere are visible under light microscopy. At the tip of the green arrow is the darkly stained A band, which is defined as the length of the myosin-thick filament. The lightly stained I band spans the orange line and contains only actin filaments. A dark line, known as the Z line, bisects each I band and attaches to the actin filaments. It can be seen at the tip of the light blue arrow. Z lines define the borders of a sarcomere. Thus, a sarcomere is composed of one A band and flanked by 1/2 of an I band on each side. Striations are a principal histologic feature of skeletal muscle. Striations are routinely evident in longitudinal sections of skeletal muscle prepared with H&E. Muscle striations appear as alternating dark and light bands. Now that we understand the structural components of the sarcomere, we know that A and I bands are responsible for skeletal muscle striations. During muscle contraction, the myofilaments of each sarcomere slide past each other and pull the Z lines towards each other. As a result, the sarcomere shortens. During this movement, the width of the A band remains constant, but both the I band and H band will shorten. We are now looking at slide 28, which is a muscle-tendon junction prepared with H and E. As I zoom in on the longitudinal section, I would like to point out the differences between the 2 tissue types in this slide. To differentiate between skeletal muscle and tendon, there are 2 factors that should be considered. The first is the shape of the fibroblast nuclei. The The cells of the tendon are compressed by extracellular collagen bundles, leaving only the nuclei visible. One can be seen here at the tip of the green arrow. These nuclei will appear as more thin and flattened than the muscle fiber nuclei. The second factor is the striation of the skeletal muscle fibers, seen here being outlined by the pointer. Collagen fibers are not striated under the light microscope. Finally, it is absolutely critical that you do not rely on color for discrimination because the staining of muscle and tendon can differ between slides. We are now looking at slide 219, which is cardiac muscle prepared with a Viegert stain. This specimen contains both cross-sectional and longitudinal cuts of cardiac muscle. As I zoom in on the cross-sectional slices of cardiac muscle cells, the nuclei become readily visible. Unlike skeletal muscle, the nuclei of cardiac myocytes are not peripherally located. Cardiac myocytes contain nuclei that are centrally located. The nuclei of a cardiac myocyte can be seen here at the tip of a yellow arrow. Also notice that each cardiac myocyte only contains one or two nuclei. We are now looking at a longitudinal section of cardiac muscle. Cardiac myocytes contain structures called intercalated discs. Several intercalated discs can be seen at the tip of the red arrows. Intercalated discs allow cardiac myocytes to act as a functional syncytium by attaching adjacent cells to each other. These attachments not only transmit the force of the contraction between cells, but also contain gap junctions, allowing the heart muscle cells to contract in tandem. In other words, these electrical impulses pass freely between cells through the gap junctions, so that the cardiac muscle functions as a single contractile unit. Some cardiac myocytes may join with two or more cells through intercalated discs, thus creating branches. Within the green circle, branching cardiac muscle fibers can be seen. Similar to skeletal muscle, cardiac muscle is organized into sarcomeres and it is striated. These striations can be seen here at the tip of my pointer. I would also like to point out the abundance of blood vessels within the cardiac tissue. Several blood vessels can be seen at the blue arrows. To help differentiate between cardiac muscle and skeletal muscle, here are some histologic features to consider. Cardiac muscle contains centrally located nuclei, while skeletal muscle contains nuclei that are located at the periphery of the cell. Cardiac muscle cells can bifurcate and form branches. Only cardiac myocytes contain intercalated discs. We are now looking at slide 218, which is smooth muscle prepared with an H&E stain. Let's examine a longitudinal section of smooth muscle. Many people, including myself, have a difficult time distinguishing between smooth muscle and dense, regular connective tissue. Smooth muscle can be identified histologically by the presence of centrally located, elongated nuclei. Smooth muscle cell nuclei often have a corkscrew appearance in longitudinal section. A corkscrew-shaped nuclei can be seen at the tip of the green arrow. This corkscrew shape is a result of the contraction of the cell during fixation and can be useful in distinguishing smooth muscle cells from fibroblasts in routine histological sections. We have moved to a cross-sectional view of smooth muscle. Each of the cells and their respective nuclei appear to be different sizes. This is because smooth muscle cells are spindle-shaped, or, as I like to picture, shaped like a football with tapering ends. Depending on where the section was taken through a cell, smooth muscle cells may appear to be different sizes. Cells will appear to be larger when the section is taken through the center of the cell, as seen here at the blue arrow. Cells will appear to be smaller when the section is taken through the edge of the cell, as seen here at the orange arrow. Recall that smooth muscle is not organized into sarcomeres, and therefore smooth muscle is not striated in its appearance. We are fortunate to have our resident vas deferentist, Kofi Money, here with us today to point out some key histological features of the vas deferens, including abundant smooth muscle.
We are now looking at slide 265, which is a section of the vas deferens, or as I like to say, The vas deferens contains 3 layers of smooth muscle, including the inner longitudinal layer, the middle circular layer, and the outer longitudinal layer. As I zoom in on a cross-section of smooth muscle, I would like to point out that the nuclei appear as a round dark structure surrounded by a lighter cytoplasm. This is indicated by the yellow arrow.
We are now looking at slide 239, which is a section of the middle esophagus prepared with an H&E stain. The esophagus contains 3 layers of muscle, including the longitudinally oriented muscularis mucosa, the inner circular layer, and the outer longitudinal layer. The outer longitudinal muscle layer of the middle esophagus contains a mixture of both smooth and skeletal muscle. The skeletal muscle fibers are considerably larger in width, appearing as rounded polygons with peripherally located nuclei. A skeletal muscle fiber is indicated by the red arrow. In contrast, many of the smooth muscle cells do not appear to have nuclei, as the plane of the section is taken through their tapering ends. When visible in smooth muscle, the nucleus is centrally located in the cytoplasm. Smooth muscle is indicated by the blue arrow. Since this is the last slide, I encourage you to pause the video and find the following as a general review: stratified squamous non-keratinized epithelium, loose connective tissue, and a blood vessel. That's a wrap for episode 6. Tune in next time for an excitatory look at nervous tissue. Thanks for watching Histo Helpers. We're here to help.